Hi everyone, this is Dr. Omar. This presentation will describe and define the threshold concept in molecular biology. And I will also list some of the features for this threshold concept and mention the significance of this threshold concept and give some examples in molecular biology. Well, threshold concepts, or sometimes they are called threshold knowledge, core concepts, or key concepts, are the most important ideas in a discipline. They are the key to a student's understanding of a discipline. These concepts permit students grasp full mastery of a discipline. They are concepts, once understood, they will transform the perception of a given subject, phenomena, or experience. They transform the way of understanding or interpreting or viewing something without which the learner cannot progress. They permit a new and previously inaccessible way of thinking about something. They allow students to make sense and integrate many previously unrelated ideas within a certain discipline. They are conceptual gateways or portals that help students overcome troublesome learning events. So, what are the features of these threshold concepts? First one is they are transformative. Transformative potential that changes the way in which the student views the discipline. Once a threshold concept is understood, a student perception and comprehension of the subject is completely altered, and a student will develop cognition of the discipline. The second feature is irreversible. Once they've been learned or done, they cannot be undone or unlearned. However, they can be modified. A third feature is integrative. The interrelatedness of idea. Once these concepts are learned, they are likely to bring together different aspects of the subject that previously did not appear to the student to be related. So they bridge concept within a discipline and potentially among disciplines. The fourth feature are that they are bounded. They are specific to a particular discipline. The fifth one is troublesome knowledge. It involves a set of knowledge which can be conceptually difficult, counterintuitive, incoherent, or even alien. So these are the major 5 features of the threshold concept. Now, why are they important? How pivotal they are? Why they are crucial for molecular biology? Because they are the foundation for comprehensive understanding of the discipline. Once mastered, they will transform the student's subsequent perception and cognition of the subject. Once concepted, they will facilitate apprehension of further related central ideas based on those concepts. They will permit constructing coherent but sequential and organized recognition of successive principle and knowledge of the discipline. They will allow integrating further acquired information based on the threshold concept, and they will enable reformulating coherent overall view of the subject or the discipline or the course or the module. They will enable and broadening venues of lateral thinking and understanding of complex facts and solving factual challenged queries, thus reaching utmost mastery of the discipline. Once a student perceived the threshold concept, they will be immersed in all scopes of the discipline, directly or indirectly. There will be those TC that stem for intercalating, overlapping, or bridging ideas, facts within a subject or between related disciplines. They are gateways to overcome learning difficulties and complex principles and informations. That's why they are important. So, what are the threshold concepts in molecular biology? So, there are certain concepts in molecular biology central to the mastery of the subject. I could come up with a few of these threshold concepts that I really found and believe they are crucial for understanding the module. The first concept is structures of the biomolecules, and when I say biomolecules, straight away it means nucleic acid structure, and nucleic acid structure two-part, DNA and RNA. DNA is well explained by Crick and Watson, DNA model presented by them, and also protein structure as one of the biomolecules. The second important concept, the central dogma, the core of the molecular biology. It's a three processes: DNA synthesis or DNA replication, RNA synthesis, and translation, protein synthesis. And the last threshold concept are the gene structure. Now, briefly give a brief description on each one of these concepts. Structure of biomolecules, as we said, RNA, DNA, and proteins. Why they are significant in molecular biology? Because they are the foundation for further understanding successive concepts and topics like replication. Replication is doubling or copying of the double-stranded DNA in vivo. Transcription is synthesis of the messenger RNA, the RNA, an intermediate template between the DNA and protein. Translation, making of proteins from the messenger RNA. So all these processes are really essential and really based on much better understanding the structure of these biomolecule, DNA, RNA, and protein. Not just that also, but the structure of the biomolecule is crucially important for understanding subsequent molecular techniques that are used to analyze and assess different biomolecules, DNA molecules, RNA molecules, and protein molecules, or gene molecules or structure. So these molecular techniques are either quantitative real-time PCR for assessing and analyzing DNA molecules, sequencing for DNA molecules, cloning in recombinant DNA molecules, and also molecular techniques for RNA analysis, quantitative real-time PCR, digital PCR, and for protein analysis for Western blot. So it's better understanding, good understanding of structure of the biomolecule will facilitate further cognition of topics within the module. The second threshold concept are the DNA model. Again, this is a sub-concept of the major concept of the major biomolecule structure. The DNA model that has been described by Crick and Watson really had important influence and better understanding of further topics. It is really important to understand the DNA structure, double-stranded DNA, and the way they anneal, so it will facilitate better understanding of in vivo DNA synthesis and copying as in replication, or in vitro DNA synthesis and copying as in PCR, polymerase chain reaction, one of the molecular method for copying specific part of a DNA within a DNA or within a genome, within a fragment of nucleic acid. It's also important for facilitate understanding the replication process, as a replication is two-stranded of DNA duplicate, and also for gene expression. Knowing the structure of the DNA and then structure of the gene will facilitate understanding how it is expressed. Also, the better understanding of the model will facilitate in vitro DNA understanding of the sequencing technique, which is an in vitro DNA synthesis and copying, to look for variation and changes and differences in the DNA template or DNA molecules or fragments. The double-stranded DNA also important, understanding it to facilitate and to good understanding of recombinant DNA technology, or recombinant DNA cloning, or genetic engineering, whatever description for it. It is also a way of in vivo DNA duplication and replication, and it's cloning and subcloning DNA. And also a better understanding of the model. DNA model will facilitate and facilitate understanding and grasp and cognition and recognizing the gene structure and gene expression. The third concept, threshold concept in molecular biology, is the central dogma. Central dogma in molecular biology, three steps or three processes. Three processes: DNA replication, gene expression, transcription, and translation. And finally, the fourth concept are the gene structure. It is very significant that the student or the learner learn the gene structure and understand it very well, as it is really a threshold concept for further understanding of the whole disciplines and subject of the molecular biology, as most of the molecular biology focus and circulate around the gene structure itself and looking at analysis of the gene itself. So by this, I hope really had covered at least threshold concept and made a clear picture or understanding of what does a threshold concept mean, the definition of it, features of it, significance of it, and I gave some examples in molecular biology. Thank you very much for listening.